Hello, and welcome to the Sleepy Curiosity channel. As you find a comfortable position, let your body start to release the weight of the day. If this gentle journey is already beginning to slow your thoughts, consider subscribing now, so these stories can keep finding you night after night. Now, let the relaxation settle into your breath. There's an organ inside your skull that's processing these words right now. Every syllable, every pause, every shift in tone. While it does that, it's also regulating your breathing, monitoring your heart rate, adjusting your body temperature, deciding in every fraction of a second what deserves your attention and what can be let go. All of it at once. All of it on less energy than a 20-watt light bulb uses. That shouldn't be possible. And yet, it's exactly what's happening right now. And the more we look at it, the more remarkable it becomes. To begin understanding the brain, you first have to let go of the idea that it's always been what it is today. The human brain didn't just appear, fully formed, out of nowhere. It's the result of a process that began so long ago that the human mind can barely grasp the scale involved. 600 million years. For reference, the dinosaurs went extinct about 65 million years ago. 600 million years ago, what existed on Earth were simple organisms, many of them living at the bottom of oceans, in a world with no accessible sunlight, no inhabited land, no plants, no animals as we know them. And it was in that ancient, strange world that something changed. A set of cells, in some primitive organism whose name we'll never know, began organizing themselves differently. Not to think. Not to feel. Just to detect a change in the environment and respond to it in a coordinated way. One cell sensed. Another reacted. The connection between them was the seed of everything that would come after. It was the first nervous system. Rudimentary. Almost absurdly simple compared to what it would become. But it was a beginning. And there's a detail that changes everything about that story. That basic principle never changed. The most fundamental function of the nervous system, detecting what's happening around it and generating an appropriate response, is the same today as it was 600 million years ago. What changed, across all that immensity of time, was the complexity built around that principle. Layer upon layer. Structure upon structure. Connection upon connection. Until it became the roughly 3-pound organ sitting inside your skull at this very moment. There's a claim that almost everyone has heard and almost everyone believes is true: we only use 10% of our brains. It's one of the most repeated ideas on the subject. And also one of the most completely wrong. Neuroimaging studies, which record brain activity in real time as people perform tasks or simply rest, show that virtually every region of the brain displays some level of activity, even during sleep. There is no 90% of the brain sitting idle while the other 10 does all the work. The origin of this myth is uncertain. It probably grew from misinterpretations of early 20th century experiments, or perhaps from a misattributed quote that got distorted over time until it became unrecognizable. What is true is that different regions have distinct functions, and they don't all work at the same intensity at the same time. But the idea that there's a vast unexplored territory of the brain waiting to be unlocked has no scientific basis whatsoever. In practice, keeping all that tissue active carries an extraordinary energy cost, and evolution doesn't typically maintain costly structures without a function. If 90% of the brain were useless, it would have disappeared over generations. What almost no one realizes is just how real and remarkable that energy cost is. The brain consumes roughly 20% of the body's total energy, despite making up only about 2% of total body mass. That means an organ that fits in the palms of two cupped hands demands a fifth of everything you eat, everything you breathe, all the glucose circulating in your blood. It's the most metabolically expensive tissue in the human body. And every neuron, every synapse, every electrical signal traveling down an axon carries a biological cost that's paid second by second, 24 hours a day, for an entire lifetime. For most of vertebrate evolutionary history, that cost kept growing because the brain kept growing. Slowly, over hundreds of millions of years, the central nervous system became increasingly elaborate. But something happened in the last 3 million years that has no parallel in any other period in the history of life on Earth. Brain volume in the lineage leading to Homo sapiens nearly tripled. In evolutionary terms, that's extraordinarily fast. A change that should have taken tens of millions of years happened in a fraction of that time. Researchers still debate seriously what drove that growth. The hypotheses aren't mutually exclusive. In fact, it's likely they all contributed in ways that fed into each other. Dramatic climate shifts in Africa during the Pliocene and Pleistocene demanded increasing behavioral flexibility. Environments were changing too fast for biological evolution to keep up, and a brain capable of learning and innovating was more valuable than fixed instincts. Diet changed too. Access to animal protein and, later, to cooked food freed up more energy to sustain such expensive tissue. And social life grew more complex. Navigating hierarchies, reading intentions, cooperating with allies and competing with rivals required increasingly sophisticated social processing. The result was a remarkable feedback loop. A larger brain enabled more sophisticated behaviors, which in turn created social pressures that favored even larger brains, which enabled even more sophisticated behaviors. A cycle that accelerated itself over millions of years, until it produced the organ that exists in every living human being today. And here's where things get strange, because the human brain is not the largest in the animal kingdom. Not even close. An African elephant's brain weighs around 11 pounds, more than 3 times ours. A humpback whale's can exceed 17 pounds. If absolute size were the determining factor in intelligence, elephants and whales would be incomparably smarter than humans. They're not. What matters more is the ratio between brain size and body size, what scientists call the encephalization quotient. And more specifically, the density and organization of certain cortical regions. What humans have that's truly unusual is an exceptionally well-developed prefrontal cortex relative to the rest of the brain. That region, located just behind the forehead, is associated with long-term planning, impulse control, and the ability to think about hypothetical situations, to imagine futures that don't yet exist and work toward them. No other animal appears to have this region as prominent, in the proportion that humans do. This starts to get more interesting when you consider that intelligence, in the way humans have it, isn't simply a matter of having more processing capacity. It's a very specific combination of abilities. Symbolic language, the capacity to represent the world through arbitrary sounds and signs that carry meaning. Accumulated cultural transmission, the ability to pass knowledge from one generation to the next, building on what came before instead of starting from scratch. And the ability to cooperate at scale with strangers, to trust and work with people you've never met, united by shared beliefs, rules, and institutions. No other species combines all three of those elements with the same intensity. And the human brain appears to have been shaped, in part, precisely to sustain that combination. But before we get to what the modern human brain can do, it's worth pausing on the story of fire. Because fire may have been one of the greatest architects of the human brain, and that idea is taken very seriously by researchers who study human evolution. The argument is elegant and supported by several independent lines of evidence. Cooking food radically transforms digestion. It makes proteins easier to break down. It makes carbohydrates more accessible. It destroys parasites and bacteria that would make raw food dangerous. And crucially, it allows you to extract far more energy from the same piece of meat or root than would be possible raw, without hours of exhausting chewing. Before fire, sustaining a large, active brain required a massive digestive system capable of processing large volumes of raw food. That digestive system also consumes energy. The competition was direct, gut versus brain. With cooked food, that competition eased. The same caloric intake could be achieved with less digestive effort, freeing up energy to sustain an ever-expanding cortex. At the same time, jaws and teeth could become smaller. There was no longer any need for dentition designed to handle hard, raw materials. And a skull that didn't need to support a massive jaw had more room to expand. It can't be stated with absolute certainty that fire caused brain growth. But the timing between more consistent fire use and the anatomical changes visible in the fossil record is something that can't be ignored. What almost no one realizes about brain evolution is that Homo sapiens wasn't the only human species to develop a large, complex brain. The Neanderthals, who lived in Europe and Asia until about 40,000 years ago, had an average brain volume slightly larger than that of modern humans. And there were at least 2 or 3 other species of the genus Homo coexisting with sapiens until relatively recent times in geological terms. Homo denisova, Homo floresiensis, possibly others not yet discovered or identified. What set sapiens apart was probably not raw brain size, but internal organization. Regions associated with language and social processing appear to have been more developed in our lineage. And there was something else, uncovered in recent decades through genetic analysis, and it fundamentally changed how we think about human history. Modern humans of Eurasian descent carry between 1 and 4% Neanderthal DNA. That means there was interbreeding. There were encounters. There were children. 2 species of humans, with similarly sized but differently organized brains, lived near each other for thousands of years, had children together, and then one of them disappeared. Only one is still here. And the reason for that difference remains one of the most intriguing open questions in anthropology. And there's a detail that changes everything when you think about the human brain at birth. Compared to most mammals, a human baby arrives in the world with an extraordinarily immature brain. A foal can stand and walk within hours. A baby chimpanzee actively clings to its mother from birth, with enough strength to hold on. A baby wildebeest needs to be running with the herd within minutes or it'll be eaten. A human infant, by contrast, is completely dependent for months, and its brain will take decades to reach full maturity. That seems like an enormous disadvantage. And in some ways, it is. But there's a deep reason for it, and it lies in a physical limit imposed by anatomy. A baby's skull has to pass through the birth canal, and that sets a ceiling on brain size at birth. The human pelvis is already unusually wide compared to other primates, an adaptation to bipedalism that created an evolutionary tension between the ability to walk upright and the ability to give birth to large-headed babies. The solution evolution landed on was remarkably elegant, outsourcing much of brain development to the postnatal period. What that means in practice is that the external environment, including social relationships, language exposure, sensory stimulation, emotional experiences, can actively shape the brain's architecture during the most critical years of development. The human brain is, of all organs, the one that most bears the mark of what it has lived through. That is both its deepest vulnerability and its most extraordinary strength. This gets more interesting when you consider what happens in the scale of weeks after conception. The brain begins forming surprisingly early. Just 3 weeks after fertilization, before most women even know they're pregnant, a structure called the neural tube is already forming in the embryo. That tube is the precursor of the entire central nervous system, the brain and the spinal cord. The closing of that tube, which occurs between the 23rd and 28th day of gestation, is one of the most critical events in all of human development. The pace of development in that early phase is almost impossible to imagine. At the peak of fetal development, the forming brain generates new neurons at an estimated rate of 250,000 per minute. 250,000 neurons per minute. Most of those neurons migrate from their region of origin to their final destinations by following precise chemical signals, a process of cellular navigation whose complexity is still the subject of intense research. Every neuron needs to find the right place, establish the right connections, at the right moments. When this process works correctly, the result is one of the most complex systems in nature. When something goes wrong at any step, the consequences can be profound and lasting. And a phenomenon that seems paradoxical happens during that development. The brain deliberately produces far more neurons than it will ever need, and then eliminates a large portion of them. This process, called neuronal apoptosis, occurs primarily during fetal development and the first years of life. It's estimated that up to half of the neurons generated during development are eliminated before birth. The logic behind this apparent waste is elegant. Neurons that successfully establish functional connections with their targets receive survival factors that keep them alive. Those that fail to establish those connections are eliminated in an orderly and precise way. It's a form of selection that ensures the final brain is composed of well-connected, functionally integrated neurons. Cell death, in this context, isn't a failure of the system. It's an essential part of building something that works. Without this precise curation, the result would be a disorganized mass of cells with no clear function. And that pruning doesn't stop at birth. The brain continues eliminating underused connections and strengthening the ones that are frequently activated throughout childhood, adolescence, and to a lesser degree across the entire adult lifespan. The architecture of your brain right now is, in part, an autobiography. It reflects what you practiced, what you learned, what you repeated, what you felt deeply enough to leave a mark. The first years of life represent a period of brain transformation with no parallel at any other stage of existence. At birth, a baby's brain weighs about 12 ounces. In the first year of life, it nearly doubles in size, reaching roughly 24 ounces. By age 3, it's already reached about 80% of its adult volume. That growth isn't primarily the result of new neurons, it's the result of an explosive proliferation of connections between existing neurons, and of the process of myelination, which coats the axons in a fatty sheath that dramatically increases the speed and efficiency of signal transmission. During that period, the brain is in a state of extraordinary openness to the environment. Sensory experiences, social interactions, language exposure, emotional bonds, all of it actively shapes which connections are strengthened and which are eliminated. That's why young children learn languages with an ease that adults can never quite replicate. The brain is literally building its architecture from what it's exposed to, and the language heard in those early years leaves physical traces in neural circuits that persist for a lifetime. Severe deprivation during that phase, lack of stimulation, neglect, exposure to chronic stress, can have lasting effects on brain architecture. Studies with children raised in institutions with severe deprivation of human interaction show measurable structural differences in regions associated with emotional regulation, attention, and language, visible on imaging decades later. A brain that doesn't receive what it needs during critical periods builds a different structure, and some of those differences persist long after conditions improve. And there's a detail that changes everything when you think about developmental critical periods. There's a window of time during development when the brain is especially sensitive to certain kinds of experience. If that experience occurs within the window, development proceeds normally. If it doesn't, certain abilities may never fully develop, even if the experience is provided later. The most studied example involves vision. If one eye of a kitten is kept closed during its first weeks of life, its critical period for visual development, the visual cortex corresponding to that eye never organizes properly. The animal develops permanent visual deficits in that eye, even though the eye itself is anatomically intact. It's not the eye that failed. It's the brain, which never received the signals it needed at the moment it was ready to build the right circuits. In humans, cases of children with untreated congenital cataracts show similar patterns. If the cataract isn't corrected during the first months of life, visual development in that eye is permanently compromised, even if surgery is performed years later. For language, critical periods are longer and more gradual, but the ease with which young children acquire languages, compared to the difficulty adults face, reflects that same biology. A child's brain isn't just a smaller adult brain. It's a system in a different state of openness to the world. What almost no one realizes about adolescence is that it has a far more concrete neurological basis than most people imagine. Adolescence is often described as a time of impulsive behavior, novelty-seeking, and questionable decision-making. For a long time, this was treated as a cultural issue, or one of psychological maturity. Neuroscience offers a different perspective. The adolescent brain is literally under construction. During adolescence, a significant reorganization of the prefrontal cortex takes place, the region associated with impulse control, planning, and evaluating consequences. This process includes an intense phase of synaptic pruning, in which less used connections are eliminated to make the system more efficient. The problem is that this reorganization isn't complete until roughly age 25. In the meantime, the limbic system, associated with emotion and reward seeking, is in full swing, sensitive, responsive, intense. The result is a temporary imbalance with real consequences. The emotional accelerator is present and fully functional. The executive brake is still being installed. It's a brain that feels a great deal and weighs things less, not by choice, but by biology. This gets more interesting when you consider what happens in early adulthood. Some cognitive abilities peak surprisingly early. Processing speed, for example, begins declining as early as age 20, a fact that surprises most people when they first hear it. Working memory, the ability to hold and manipulate information in mind over short periods, peaks around age 25 to 30. But other abilities follow completely different trajectories. Vocabulary and accumulated semantic knowledge grow until the 60s or 70s. Crystallized intelligence, the ability to use accumulated knowledge and experience to solve problems, remains stable or even increases in middle age. The capacity to regulate emotions, to put situations in perspective, to not overreact to setbacks, those abilities tend to improve with experience and maturity. Brain aging isn't simply a process of uniform decline. It's a reorganization in which certain speeds slow down while certain depths grow. And there's something many people don't know about brain aging. The rate of that process varies enormously between individuals, far more than aging in other organs. Two kidneys of the same chronological age tend to be in comparable condition. Two brains of the same chronological age can be in radically different states. Factors like regular physical exercise, ongoing cognitive engagement, sleep quality, diet, and social connection have well-documented associations with the preservation of brain function over time. A fundamental concept in this area is cognitive reserve. Brains with a greater number of connections and more complex neural networks can tolerate more damage before showing functional symptoms. People with higher levels of education, greater intellectual engagement throughout life, and richer social networks tend to show symptoms of dementia later, even when post-mortem analyses reveal comparable degrees of physical damage to brain tissue. Reserve doesn't prevent the damage, but it increases the brain's ability to compensate for it, like a building constructed with more support columns than necessary, which stays standing even when some of them fail. And there's a detail that changes everything when you start to understand how the brain is organized on the inside. The most basic division separates the brain into three major regions that reflect, in part, the evolutionary history of the organ, the layers that were added over hundreds of millions of years. The brainstem, at the base, controls automatic vital functions, breathing, heart rate, blood pressure, sleep-wake cycles. It's the oldest region, the most evolutionarily conserved, shared with virtually all vertebrates. Damage to it is potentially fatal, because it sustains the functions without which no other function is possible. The cerebellum, at the back and bottom, coordinates movement and balance with remarkable precision. For decades, it was seen primarily as a motor module. that view is being significantly revised. More recent studies show the cerebellum has extensive connections to cortical regions associated with thought, language, and emotion. People with cerebellar lesions often show not only motor difficulties, but also subtle changes in cognition and emotional processing. And there's a number that surprises almost everyone. The cerebellum contains approximately 69 billion neurons, more than half of all the neurons in the human brain, compressed into a structure that occupies only 10% of the total volume. An extraordinary density of processing in an extraordinarily compact space. And the brain proper, the telencephalon, with its two hemispheres and folded cortex, is the structure that grew the most in mammals and that, in humans, visually dominates the entire cranial anatomy. It's where much of what we associate with thought, perception, language, and complex emotions resides. And within it are smaller structures of enormous importance, regulating fundamental aspects of human experience. The hypothalamus, with a volume of just about 4 cubic centimeters, functions as a control center that integrates signals from the environment, the body, and the brain itself to regulate temperature, hunger, thirst, sleep, reproductive behavior, and stress response. Its influence extends far beyond what its size would suggest. It controls the pituitary gland, which in turn regulates hormone release throughout the entire body. A structure the size of a grape governs part of what the whole body feels, needs, and does. Small lesions in this region can cause dramatic changes in behavior and physiology, disruptions to temperature regulation, appetite, sleep, sexual behavior, which gives some sense of how much power is concentrated in this unassuming structure. What seems simple isn't, either, when you examine the individual neuron. A neuron is a cell with an unusual shape, and that shape isn't accidental, it reflects exactly the function it performs. Every neuron has a central body where the nucleus sits, and two types of extensions. The dendrites, usually numerous, branching, resembling tree limbs, function as receiving antennas, capturing signals coming from other neurons. The axon, usually single and longer, carries the electrical signal away from the cell body, toward other neurons, muscles, or glands. The length of axons varies in an almost absurd way. Some measure fractions of a millimeter, connecting neighboring neurons within the same brain region. Others are extraordinarily long. The motor neurons that control the muscles of the feet have axons that run from the spinal cord all the way to the lower extremities, reaching more than 3 feet in length in tall adults. A single neuron can therefore have dimensions that range from the microscopic to the macroscopic. It's as if a single cell exists simultaneously at completely different scales. Two neurons never directly touch. Between them there's always a gap, the synapse, between 20 and 40 nanometers wide. A human hair is about 70,000 nanometers in diameter. The space between two neurons is therefore almost 2,000 times smaller than a single strand of hair. In that nearly non-existent space, the communication between the cells that govern every thought, every movement, every emotion occurs through chemical molecules called neurotransmitters. They're released on one side, cross the gap, and lock into specific receptors on the other side. The entire process takes fractions of a millisecond, and each neuron can have between 1,000 and 10,000 synaptic connections with other neurons. Multiply that by the total number of neurons, 86 billion, and the human brain is estimated to contain somewhere between 100 trillion and 500 trillion synapses. 500 trillion. It's at that scale of complexity that everything we call the mind emerges. Not in individual neurons. Not in isolated regions. In patterns. In rhythms. In simultaneous conversations among billions of cells distributed across a system that fits inside a show box. This gets more interesting when you understand how the electrical signal travels through a neuron. It works in a binary way. A neuron either fires or it doesn't. There's no middle ground in terms of the individual signal. That firing is called an action potential, and its amplitude is always the same for a given neuron, regardless of the intensity of the stimulus that triggered it. It's the all-or-nothing principle. A light touch and an intense pain produce the same type of electrical signal in each individual neuron. How, then, does the brain tell the difference between a whisper and a deafening noise? Between a gentle touch and a burn? The answer lies in the frequency and pattern of firing, not the amplitude of each signal. A stronger stimulus causes a neuron to fire more times per second and recruits a greater number of neighboring neurons. It's in the combination of frequency, timing, and activation patterns across entire populations of neurons that information is encoded. The difference between one musical note and another, between a familiar face and a stranger's, between a moment of peace and a moment of terror, it's all in the patterns, not the individual signals. And there's a detail that changes everything about how those patterns form and stabilize. There's a principle that governs the strength of connections between neurons and that underlies learning and memory. Formulated by neuropsychologist Donald Hebb in 1949, it can be simply stated, neurons that fire together wire together. When two neurons are repeatedly activated at the same time, the synapse between them strengthens, making it more likely that activating one will lead to activating the other in the future. This process is called long-term potentiation, and it's considered the cellular mechanism underlying memory formation and learning. The reverse also occurs. Connections that are rarely used tend to weaken and eventually disappear. That's why regularly practiced skills become more automatic over time, and why knowledge that isn't revisited tends to fade. The brain literally reorganizes its physical connections based on what gets used. Every time you practice something, Every time you repeat a mental pathway, you're changing the biology of your brain. Not metaphorically. Structurally. What almost no one realizes about neurons is that they're extraordinarily long-lived cells. Most of the neurons a person has at 30 have been present since the beginning of life, some since before birth. Unlike skin cells, which are replaced every few weeks, or blood cells, which live for months, neurons can survive for decades and accumulate a unique biochemical history. A neuron you have today may have been formed before you were born and remained active, connected, functioning, throughout your entire life up to this moment. That longevity has interesting implications. It means neurons need extremely efficient maintenance systems to stay functional over decades. It also means damage to neurons, caused by trauma, lack of oxygen, or neurodegenerative diseases, tends to be permanent in most brain regions. The most studied exception is the hippocampus, where there's evidence of new neuron generation throughout adult life, though the magnitude and functional significance of that process in humans is still vigorously debated among researchers. Beyond neurons, the brain contains an equally numerous population of cells called glial cells. The name comes from the Greek word for glue, reflecting the old idea that they served only as structural support for the real neurons. That view has been completely revised in recent decades. Glial cells play active and varied roles, and understanding the brain without them is like trying to understand a city by looking only at the roads and ignoring everything else. Astrocytes regulate the chemical environment around neurons and modulate synaptic transmission, actively participating in conversations between neurons, not just observing. Oligodendrocytes produce the myelin sheath that coats the axons. Microglia function as the brain's immune system, detecting damage and responding to infections and inflammation. A recent and intriguing discovery is that human astrocytes are significantly larger and more complex than those of other mammals. When human astrocytes were transplanted into mouse brains in laboratory experiments, the animals showed measurable improvements on learning and memory tasks. The support cells of the human brain, it turns out, do something that the support cells of other animals don't do in quite the same way. This gets more interesting when you consider how the brain is supplied with blood. Despite making up only 2% of body mass, the brain receives roughly 15% of the total cardiac output. To sustain that demand, there's an extraordinarily dense vascular network. The total length of blood vessels in the brain, adding arteries, veins, and capillaries, reaches about 400 miles. 400 miles of vessels inside an organ the size of a small melon. A unique feature of that vascularization is the blood-brain barrier, a filter formed by the cells lining the brain's capillaries. This barrier is selective. It allows the passage of oxygen, glucose, and some essential molecules, but blocks most of the substances present in the blood, including many medications. This protects brain tissue from potentially harmful agents, but it also represents one of the greatest challenges in developing treatments for neurological diseases. Getting to the brain, even with the right therapeutic molecule, requires crossing a boundary that evolved over hundreds of millions of years to keep the outside world out. And there's a detail that changes everything about how pain works. The brain doesn't feel pain. Brain tissue itself has no pain receptors, the so-called nociceptors. That's why brain surgeries can be performed with the patient awake, which sounds unsettling but is an established practice in certain clinical situations, since it allows the surgeon to monitor neurological functions in real time while operating. The headaches everyone knows don't come from brain tissue, but from surrounding structures, the meninges, the blood vessels, the muscles of the scalp and neck. Migraines involve the activation of pain pathways associated with the meninges and intracranial vessels. The organ that processes all of the body's pain is, itself, incapable of feeling pain. There's something almost poetic in that asymmetry. The center that receives all of the organism's messages of suffering can't be directly reached by any of them. For a long time, the brain was considered essentially static in adulthood, a structure that formed in childhood and then remained relatively fixed. That view began to change significantly starting in the 80s and 90s. With growing evidence that the adult brain retains a considerable ability to reorganize itself in response to experience. The phenomenon was given the name neuroplasticity. It occurs at several levels simultaneously. Connections between neurons can strengthen or weaken with use. New connections can form. In some specific regions, new neurons can be generated even in adulthood. And entire regions can expand or shrink their cortical representation in response to changes in what is practiced or lost. Studies with professional musicians show measurable structural differences in the brain compared to non-musicians, greater volume in motor and auditory regions, a thicker corpus callosum. In blind people who use Braille, the cortex dedicated to the fingertips grows substantially. In people who lose a limb, the area previously dedicated to it begins to be recruited by neighboring regions. The map of the body in the brain is dynamic, responsive, alive. What almost no one realizes about neuroplasticity is that it's not just a capacity for recovery after damage. It's the brain's normal mode of operation. With every new experience, every skill practiced, every intense emotion lived, every full night of sleep, the brain is reorganizing itself. Quietly. Without warning. Without the conscious mind noticing what's being built or undone. The person you are tomorrow will have a brain that's literally different from the one you have today, in molecular and synaptic details that no current instrument could measure, but that are real. This gets more interesting when you think about memory. For a long time, it was compared to a recording, something that captures events and stores them for future retrieval. That analogy is convenient, but fundamentally wrong. Every time a memory is accessed, it isn't simply played back. It's actively reconstructed, from fragments distributed across different brain regions, reassembled in the moment of remembering from pieces stored in distinct locations. And that reconstruction process makes memory surprisingly malleable. Details can be altered. Gaps are filled in with plausible information based on what makes sense, not necessarily on what actually happened. The context at the moment of retrieval influences what's remembered and how. The psychologist Elizabeth Loftus demonstrated over decades of research that false memories can be implanted with relative ease. In controlled experiments, participants came to remember with conviction events that never happened, like getting lost in a shopping mall as a child, like witnessing an accident they didn't see, like having done things that other people had done, after subtle suggestions and carefully constructed contexts. This has implications that go far beyond academic psychology. Eyewitness testimony, treated by the legal system as privileged evidence, is a brain construction subject to all the imperfections, biases, and vulnerabilities of any other memory. The study of false memories has directly contributed to the review of criminal cases in which people were convicted based on testimony that turned out, decades later, to be biologically impossible to trust. Memory isn't a single thing. It's a family of distinct systems that operate in different ways and depend on different brain structures. The most fundamental distinction separates declarative memory from non-declarative memory. Declarative memory involves facts and events that can be consciously accessed and verbalized: the name of a capital city, what happened yesterday, the face of someone you know. Non-declarative memory involves skills, habits, and conditioned responses that express themselves in behavior without requiring conscious access: riding a bike, typing without looking at the keys, the grammar of your native language. That distinction isn't just theoretical. It's anatomically real. Patient HM completely lost the ability to form new declarative memories after the removal of his hippocampus. But he continued learning new motor skills normally, without ever being aware that he was learning. Each training session began from scratch for him, with no memory of the previous ones, but his performance improved consistently and measurably. The brain was learning, even though the conscious mind didn't know it. And that dissociation reveals something profound. The part of us that thinks about what it knows is only a fraction of the system that actually holds what we've learned. And there's a detail that changes everything about how memory consolidates. There's a critical window for that, one that opens every night, during sleep. The consolidation of memories, the process by which recent experiences are stabilized and integrated into long-term storage, depends heavily on sleep, and different sleep phases appear to play distinct roles in that process. Slow-wave sleep, predominant in the early part of the night, seems especially important for the consolidation of declarative memories, facts, events, learned information. During that deep sleep, the hippocampus appears to replay neural activity patterns that occurred during the day, as if rehearsing recent experiences for the neocortex, which gradually integrates them into existing knowledge. REM sleep, more abundant in the final hours before waking, appears to favor the consolidation of procedural and emotional memories. Depriving someone of sleep after a learning experience significantly impairs retention. Sleeping between a study session and a test measurably improves performance. Sleep is not a pause in learning. It's part of the process. Perhaps the most important part, because it's exactly when the external environment stops competing for the brain's attention, and it can finally organize in peace what it accumulated throughout the day. What almost no one realizes about forgetting is that it's often not a failure. It's an active and adaptive process, not passive deterioration. Remembering everything indiscriminately would be, in practice, debilitating. Rare cases of people with highly superior autobiographical memory illustrate this in a concrete and unsettling way. These individuals remember in extraordinary detail virtually every day of their lives from a certain age onward. They make no deliberate effort, the memories are simply there, accessible with a vividness that people with typical memory rarely experience even for recent events. Far from being a pure advantage, many describe the condition as overwhelming. They relive negative memories with the same intensity as positive ones. They struggle to focus on the present because the past never falls silent. They're dominated by an archive that never fully closes. Selective forgetting appears to be what allows the brain to keep useful memories accessible without being overwhelmed by the accumulated weight of everything it has ever experienced. Forgetting isn't always a loss. Sometimes it's exactly what the system needs to keep functioning. This gets more interesting when you consider the effect of emotion on memory. The amygdala, an almond-shaped structure in the temporal lobe, is at the center of that interaction. When an experience is emotionally intense, the amygdala signals to the hippocampus that memory deserves priority. Consolidation happens faster and more robustly than for equivalent neutral events. That's why emotionally charged events, an accident, devastating news, a moment of intense joy, tend to be remembered with far more vividness and detail than neutral events of comparable complexity. That prioritization makes deep evolutionary sense. Remembering with precision situations that generated intense fear or pleasure is adaptive, knowing which environments are dangerous, which behaviors were rewarded, which people can be trusted. The problem arises when the system is activated by trauma. The same machinery that makes emotional memories lasting is what makes traumatic memories so persistent, so resistant to forgetting, so capable of intruding on the present without warning. The strength that was meant to protect becomes, in those cases, the very thing one most needs protection from. And there's a detail that changes everything about how the amygdala operates in real time. It receives visual signals through a subcortical pathway that arrives before information is processed by the visual cortex. That means the amygdala can evaluate a potential threat and initiate an alert response before there's any conscious awareness of the stimulus. That's why a person can flinch at something that looks like a snake, and only afterward consciously realize it was just a stick. The fear response happened before consciousness had time to process what it was seeing. That mechanism is extraordinarily efficient for survival in an environment where real threats could appear without warning. The cost is a tendency toward false alarms. And in modern life, where most threats aren't snakes but difficult emails and tense conversations, this system calibrated for the savannah keeps firing with the same urgency. The heart rate climbs. Breathing shifts. Muscles tighten. Everything prepared for a physical confrontation that will never happen. The brain does what it was shaped to do, just in the wrong environment. Chronic stress, in that context, is one of the most well-documented forms of brain damage that doesn't show up on standard imaging. Prolonged exposure to cortisol, the central hormone of the stress response, has measurable and harmful effects on the brain. It reduces hippocampal volume, impairs memory and learning, increases amygdala reactivity, and weakens connections in the prefrontal cortex. Precisely the regions that allow us to regulate emotions, make considered decisions, and plan for the future are compromised by prolonged exposure to stress. And there's something even more disturbing in that chain. Chronic stress doesn't just damage the regions that would help manage it. It increases the reactivity of the amygdala, making the brain more sensitive to new threats, quicker to trigger alarm responses, harder to calm down. It's a cycle that feeds itself, and one that has only been well understood in recent decades. Chronic stress doesn't just harm the body. It literally reorganizes the brain in ways that perpetuate vulnerability to stress itself. What seems simple isn't. Dopamine, the neurotransmitter most talked about in popular culture, is often described as the pleasure neurotransmitter. The reality is more precise and, paradoxically, more revealing. Studies over the past 30 years consistently show that dopamine is more associated with anticipation and motivation than with pleasure itself. It's released when the brain predicts a reward, not necessarily when it receives one. That detail has profound implications and explains a great deal of human behavior. The dopamine system is more activated by the possibility of reward than by a guaranteed reward. Unpredictable rewards, those that arrive sometimes, at random, are more addictive than certain ones. The brain doesn't get used to uncertainty, it keeps responding, keeps seeking, keeps waiting. Gambling, social media with unpredictable notifications, variable reward systems in apps designed for engagement, all of them exploit exactly that mechanism. The brain isn't seeking pleasure. It's seeking the promise of pleasure. And it keeps seeking even when that promise rarely comes through. This gets more interesting when you consider the relationship between emotion and reason. For a long time, they were treated as opposing forces. Emotion as something that interferes with rational thinking. Reason as what separates us from merely instinctive responses. Neuroscientist Antonio DiMaggio turned that perspective on its head with studies that still challenge the intuitions of many people. He studied patients who had suffered damage to the ventromedial prefrontal cortex, a region that integrates emotional processing and decision-making. These patients retained intact their capacities for logical reasoning, memory, and language. They could discuss abstract problems with sophistication. They could analyze situations hypothetically. But their practical lives fell apart in surprising ways. They were incapable of making simple everyday decisions, choosing between two restaurants, settling on a meeting time, without deliberations that lasted hours and frequently reached no conclusion at all. And their personal and financial choices were often disastrous. DiMaggio's hypothesis was elegant. Emotions function as rapid evaluative markers that guide decision-making, especially in complex situations where pure reasoning would be too slow or simply insufficient. Without access to those emotional markers, decisions don't become more rational. They become paralyzing or arbitrary. Emotion doesn't interfere with reason. It makes reason possible. And there's a detail that changes everything about how the brain perceives the world. What we call seeing has very little to do with passively capturing the outside world. The brain doesn't receive a complete image and display it internally. It actively reconstructs reality from fragments of incomplete information, using expectations, memories, and inferences to fill in what's missing. Perception is a construction, not a recording. And the difference between those two things is far greater than it seems. The retina has a blind spot, a region with no photoreceptors, where the optic nerve connects to the eye. Any object that projects an image onto that spot is invisible to that eye. And yet, no one perceives a black hole in their visual field. The brain simply fills in that space with what makes sense based on the surrounding context. That simple example reveals something broader and deeper. What you experience as visual reality is, in large part, a hypothesis the brain has constructed about what's probably there. Seeing is, fundamentally, a creative act. It always has been. What almost no one realizes is that there's a delay between the moment something happens in the world and the moment the brain becomes conscious of it. Visual signals take roughly 100 milliseconds to travel from the eye to the visual cortex and be processed enough to generate conscious perception. Signals from other sensory modalities arrive at slightly different times. To create a coherent, synchronized perceptual experience, the brain has to align information that arrived at distinct moments, and it does this retroactively, recalibrating the timeline of events. The result is that conscious perception is always, to to some degree, a slightly delayed and edited reconstruction of the present. That now you experience isn't the instant things happen. It's a narrative constructed with a small delay, polished to appear continuous and immediate. The sense of inhabiting a flowing present is itself a production of the brain. A functionally indispensable illusion, without which every experience would be a disconnected fragment rather than a story that makes sense. And there's something remarkable the brain does when it isn't performing any specific task. For a long time, it was assumed that when a person isn't focused on something, the brain simply reduces its activity. Neuroimaging showed the opposite. When the brain isn't focused on an external task, a specific network of regions becomes more active, not less. That network was called the default mode network. It's active during daydreaming, reflection on the past, projections about the future, thoughts about other people, and the elaboration of personal narratives. Researchers believe it plays an important role in memory consolidation, long-term planning, and the construction of personal identity, that continuous sense of being yourself over time. The brain's version of doing nothing is actually a highly organized state of processing. The moments when the mind wanders aren't empty. They're when the system integrates, synthesizes, and builds connections that focused attention sometimes can't. This gets more interesting when we consider altered states of consciousness and what they reveal about how consciousness normally works. General anesthesia is one of the most widely used medical tools and, paradoxically, one of the least understood in its fundamental mechanisms. Decades after its routine use, anesthesiologists still don't have a complete model of how anesthetic agents produce loss of consciousness. What neuroimaging has revealed is that under anesthesia, the brain doesn't simply shut down uniformly. Many brain regions continue to be individually active. What's lost is the coordinated communication between them. The fragmented brain, active in parts, but unable to integrate, loses consciousness. This suggested something profound. Consciousness may not depend on any specific region of the brain, but on the degree of information integration between regions. When that integration falls below a certain threshold, even if individual neurons keep firing, the subjective experience disappears. And there's a detail that changes everything about how we dream. During REM sleep, the brain is highly active, with activity patterns that instruments sometimes confuse with the waking state. But the body is essentially paralyzed. The voluntary muscles receive active inhibitory signals that create an almost complete absence of muscle tone. It's not a side effect of sleep. It's a deliberate protective mechanism that prevents the body from physically acting out the dreams. The most dramatic evidence of this paralysis's function comes from a condition in which it fails: REM sleep behavior disorder. People with this condition shout, gesture, kick, and even injure themselves during REM sleep, physically acting out the content of their dreams. Beyond being dangerous, This condition revealed something clinically important. It's considered an early marker of neurodegenerative diseases like Parkinson's and Lewy body dementia, often appearing years or decades before the main motor or cognitive symptoms. Sleep tells a story about the brain's health long before any other signal appears. Dreams remain one of the most studied and least understood phenomena in all of neuroscience. An influential hypothesis proposes that REM sleep functions as a kind of nightly therapy, a state in which emotionally charged memories are reprocessed in an environment with reduced levels of the neurotransmitter associated with stress. This would allow the emotional content of memories to be integrated without the intense affective charge of the original experience. People with post-traumatic stress disorder frequently show disruptions in that nightly reprocessing, recurring nightmares, waking in the middle of REM sleep, fragmented sleep overall. And there's research showing that interrupting REM sleep can perpetuate the emotional intensity of trauma. While asleep, the brain tries to heal what it couldn't resolve while awake. Sleep isn't a pause in emotional life. It's where some of the most important emotional work happens, quietly, without conscious supervision, while the body rests. What almost no one realizes about sleep deprivation is the real scale of the impairment it causes. After 17 hours without sleep, cognitive performance deteriorates to a level comparable to someone with a blood alcohol content of 0.05%, close to the legal limit for driving in many countries. After 24 hours without sleep, the impairment is equivalent to roughly 0.1% blood alcohol. And unlike alcohol intoxication, which produces a clear subjective sense of being altered, sleep deprivation dulls metacognition, the ability to evaluate one's own cognitive state. People chronically deprived of sleep adapt to the feeling of tiredness and come to consider it normal, while continuing to show measurable deficits in attention, working memory, emotional regulation, and decision-making. The perception that I'm fine with 5 hours of sleep is itself a symptom of the impairment. The sleep-deprived brain can't accurately assess how deprived it is, which creates the conditions for deprivation to continue and deepen without the person realizing the extent of the problem. This gets more interesting when you consider the state that exists on the threshold between sleep and wakefulness. Hypnagogia, the state that occurs in the transition from wakefulness to sleep, is characterized by a mixture of fragmented rational thinking and involuntary, vivid images, sounds, or sensations. The brain is partly in the logic of waking consciousness and partly in the logic of sleep, and the results of that mixture can be extraordinary. Several creators and thinkers throughout history reported deliberately using this state as a source of ideas. Thomas Edison, Salvador Dali, and Nikola Tesla are often cited as practitioners of techniques to remain in that threshold without slipping fully into sleep. From a neurological standpoint, the hypnagogic state is characterized by a mixture of alpha waves, associated with relaxed wakefulness, and theta waves, associated with the early stages of sleep, that creates unique conditions for unusual associations and non-linear thinking. It's the state in which conscious control relaxes just enough to let unlikely connections emerge. And there's a phenomenon that many people experience but few know has a name and a documented neurological basis. Sleep paralysis occurs when the muscle atonia mechanism of REM persists while consciousness returns, leaving a person fully awake and aware, but unable to voluntarily move any part of their body. The episode typically lasts anywhere from a few seconds to a few minutes, but can feel much longer, especially because it's frequently accompanied by vivid hallucinations, the sense of a presence in the room, figures, sounds, a feeling of pressure on the chest. Cultures around the world, with no communication between them, developed remarkably similar supernatural explanations for that experience: night demons, witches sitting on the chest, visitors from other worlds. The universality of those narratives reflects the universality of the underlying biological experience. Today it's understood to be a benign phenomenon caused by a transient desynchronization between the systems that control the state of consciousness and those that control muscle tone. But the fact that so many different cultures created the same stories to explain the same phenomenon says something about how the human brain responds to the inexplicable. It always creates a narrative. Always. And there's a detail that changes everything about how consciousness is constructed. Cotard syndrome, the delusion of negation, leads the patient to genuinely believe they are dead, that they don't exist, or that they've lost essential internal organs. It's not an expression of emotional suffering. It's a literal and unshakable belief that resists rational argument, showing the patient that their heart is beating, that their blood is circulating, that they obviously exist and are right there. None of it convinces. The brain has reached a conclusion and holds to that conclusion against all evidence. Neuroimaging studies show dramatic reductions in the metabolism of regions associated with the integration of bodily self-perception and the sense of agency, the experience of being the author of one's own actions and experiences. The current hypothesis is that the syndrome results from a disconnection between the systems that process sensory information and those that generate the subjective feeling that that information belongs to a living, present self. When that integration fails completely, the brain may reach the conclusion, incoherent with the fragmented information it's receiving, that the self simply no longer exists. The sense of existing isn't a given. It's constructed. And when the construction fails, existence disappears. What almost no one realizes about consciousness is that it may not be where we imagine it is. The question of where consciousness resides in the brain, whether it resides somewhere specific, or whether it emerges from patterns distributed across the whole network, remains without a definitive answer. One of the most unsettling discoveries about human perception is that visual, auditory, and motor processing is happening all the time in the brain that never reaches consciousness. Patients with blind sight, which occurs after damage to the primary visual cortex, report seeing nothing in part of their visual field. Yet when asked to guess where an object is positioned in that blind region, they answer correctly at a rate far above chance. When made to navigate around obstacles in that region, they often do so correctly without knowing why. What this reveals is that visual processing in the brain follows multiple parallel pathways. The pathway through the primary visual cortex appears to be necessary for the conscious experience of vision. But other subcortical pathways continue processing visual information and guiding behavior, without any of that reaching consciousness. Blind sight suggests that the brain knows things the conscious mind doesn't. And it acts on that invisible knowledge. Consciousness, it seems, is only the illuminated surface of a far vaster processing that takes place in the depths. This gets more interesting when we consider empathy and what it reveals about how the brain processes other minds. One of the discoveries that generated the most excitement was the identification of mirror neurons, first described in monkeys in the 90s. These neurons fire both when the animal performs an action and when it observes another individual performing that same action. As if the brain were internally simulating what the other is doing, rather than simply registering it as external information. In humans, neuroimaging studies show that observing someone in pain, fear, or pleasure activates brain regions that overlap with those activated when experiencing those emotions directly. Empathy appears to involve an internal simulation of another state. The brain literally rehearses the other person's experience using its own neural representations. That simulation isn't identical to direct experience, but shares part of the same neural infrastructure. Feeling with another person isn't just an emotional disposition. It's a neurological process. And when it works well, it creates the foundation for everything we call human connection. And there's a detail that changes everything about how decisions are made. There's a systematic tendency to evaluate losses as more intense than equivalent gains. The pain of losing something is psychologically worth about twice as much as the pleasure of gaining something of equal value. That asymmetry has an identifiable neurobiological basis. Losses engage the amygdala and threat response regions more strongly, while gains predominantly activate the dopaminergic reward system. From an evolutionary standpoint, that asymmetry makes deep sense. The costs of losing resources essential to survival were historically far more severe than the benefits of additional gains. An ancestor who didn't risk their only food source for a chance at more food survived longer than one who did. The problem is that this system calibrated for scarcity now operates in environments very different from those in which it was shaped. And most of what behavioral economics documents as irrationalities in human decision-making are, in fact, direct consequences of a brain solving the wrong problem, surviving on a savanna, not navigating a financial market or a list of insurance options. What seems simple isn't when it comes to free will. Neuroscience entered this age-old philosophical question with tools that philosophy didn't have. In 1983, neuroscientist Benjamin Libet showed, using brain activity recordings, that a wave of neural activity, called the readiness potential, proceeds by about 550 milliseconds the moment a person reports having consciously decided to move their hand. The awareness of the decision arrived after the brain had already initiated the process. The experiments generated decades of philosophical and scientific debate that continue to this day. Critics pointed out serious methodological limitations. Later studies produced more nuanced results, suggesting that the readiness potential may represent non-specific motor preparation rather than a specific decision. But the central question remains: if unconscious brain processes proceed and possibly determine what we called conscious decisions, what's left of the concept of voluntary agency? Neuroscience hasn't resolved that question, but it made it empirically investigable in ways that philosophy alone never could. And that may be the most unsettling gift science has given to humanity, the possibility of rigorously investigating the nature of what we are. There's a specific mental state that combines high concentration, total engagement, and a loss of the sense of time, described by psychologists Mihaly Csikszentmihalyi as flow. People who experience it often describe it as one of the most satisfying states possible. Artists, athletes, programmers, musicians, many report that the best experiences of their lives happened in that state. And neuroscience has begun to map what happens in the brain when it emerges. One of the most consistent findings is a reduction of activity in the prefrontal cortex during the flow state, a phenomenon called transient hypofrontality. The inner critical voice and the constant monitoring of one's own performance depend heavily on the prefrontal cortex. When that region reduces its activity, execution flows more automatically and with less interference from self-awareness. At the same time, there's increased coherence between brain regions involved in the specific task. The flow state appears to be a highly efficient processing state, focused, coordinated, and temporarily freed from the weight of excessive conscious monitoring. The paradox is that exerting less effort at control can result in better performance. This gets more interesting when you consider what psychedelic substances reveal about the brain. For decades, they were practically banned from scientific research. Over the last 2 decades, a wave of clinical studies at universities like Johns Hopkins, Imperial College London, and NYU reopened that field with rigorous methodology. What neuroimaging studies show is that psychedelics produce a significant increase in connectivity between brain regions that normally have little direct communication, while reducing activity in the default mode network. The current hypothesis is that the psychedelic experience corresponds to a state of higher brain entropy, more possible states of activity, more unlikely connections, less dominance of habitual patterns. The brain, in that state, behaves in ways it normally suppresses. This may explain both the subjective effects of novelty and boundary dissolution, and the therapeutic potential that clinical studies have documented for treatment-resistant depression, anxiety associated with terminal illness, and post-traumatic stress disorder. A brain that opens itself to unlikely connections seems, in certain contexts and under carefully controlled conditions, to be able to solve problems that the brain in its habitual state could not. And there's a detail that changes everything about how the gut microbiome relates to the brain. The gut sends more signals to the brain than it receives from it. The vagus nerve, one of the largest communication pathways in the nervous system, carries the majority of its fibers upward, from the abdomen to the brain. And the trillions of bacteria inhabiting the digestive tract produce neurotransmitters, regulate inflammation, and influence mood in ways that research over recent decades is only beginning to map with precision. Animal studies have shown that changes to the gut microbiome produce measurable changes in behavior and stress response, and that transferring the microbiome from anxious animals to animals without a microbiome transfers anxious behavior patterns along with it. In humans, associations between microbiome composition and conditions like depression, anxiety, and Parkinson's disease have been documented. The idea that mental state could be influenced by bacteria in the gut would have seemed implausible 30 years ago. Today it's one of the most active frontiers in neuroscience. And it raises a question that still has no answer. Where exactly does the brain end and the rest of the organism begin? What almost no one realizes about the project of mapping the human brain is the scale of what still remains to be done. The human connectome, a complete map of all neural connections, would involve mapping somewhere between 100 trillion and 500 trillion synapses. In 2023, researchers completed the map of a tiny fragment of mouse cerebral cortex, a 1 cubic millimeter cube of tissue, and the result occupied 1.4 petabytes of data. 1 cubic millimeter of tissue from a mouse. Extrapolating that to the entire human brain gives a dizzying sense of what's inside every human skull. And even if that map were completed, it's not clear that would be enough to understand the brain. Structure is not function. Knowing where the connections are doesn't automatically reveal what emerges when those connections activate together, in patterns that change every millisecond, in response to an environment that never repeats itself in exactly the same way. The brain may be the only object in the universe complex enough to defy any system of representation that attempts to capture it completely. And here we arrive at the deepest question. It's not about a specific disease, an extraordinary ability, or a particular neural mechanism. It's about what the brain really is. The organ that evolved to survive on African savannas now produces literature, music, mathematics, and science. The same system that was shaped by natural selection to make rapid decisions in immediate environments now tries to understand itself, which represents a level of self-reflexivity without known precedent in any other natural system. We know a great deal about neurons, synapses, regions, and networks. but how patterns of electrical and chemical activity in biological tissue produce the experience of being someone, with memories, emotions, perspectives, and the feeling of inhabiting a continuous present, remains unanswered. Not for lack of effort or methodology, but because this may be the hardest question any cognitive system has ever tried to answer about itself. And the fact that the human brain is capable of formulating that question with enough clarity to recognize its own difficulty is, in itself, one of the most extraordinary phenomena science has ever encountered. You're not just thinking with a brain. You are the moment the material universe arrived at the point of asking what it is. There's something deeply calming in that, when you stop to feel it. All the weight we carry, the worries, the decisions, the regrets, the plans, all of it passes through this 3-pound organ that never fully rests, that cleans itself while you sleep, that consolidates what you lived while you weren't looking. There's a deep intelligence in the body that doesn't need your supervision to do what it needs to do. Now, as the day grows more distant, as your breathing slows, the system that processed all of this begins its most important work. The silence is part of the process. The rest is part of the building. And as you drift into a deep and restorative sleep, the glymphatic system begins its quiet work. The hippocampus softly whispers to the neocortex what happened today, the amygdala slowly lowers its guard, and the prefrontal cortex, which worked all day long, finally rests. Remember to subscribe, so these stories can keep finding you, night after night. Sleep well, and sweet dreams.